And this, oh yeah, thanks, Matt. And I have a couple computers going. Okay, and this is the live portion of my IPM training. I have there's a couple of videos online on YouTube, and I sent that link in an email yesterday, and that includes some information on different strawberry pests and berry production systems. So you can check that out as well. And if you have any questions about those, we can cover those today as well. Okay, so I'm going to cover a few things today: scout safety, strawberry disorders, strawberry insect pests, strawberry diseases, a few raspberry pests. I'll spend a little bit of extra time on Spotawing Drosophila because it's such a significant pest for berry producers. And then I'll give you a few resources in addition to the ones that I sent yesterday. Okay, first, I want to try launching a poll just to get to know who's joining us today. So the first question, let me launch it. There we go. So the first question is, what is your role in the berry industry? Who's joining us today? Are you a grower, farm employee, summer student, research or government, from an input or retailer, or other? So just looking to get a feel for who's joining us. I'll give it a couple more seconds. Okay, and there are results. So you can see that we have a mix of people joining, which is great. Good to have folks joining from all different parts of the industry. So thanks for joining, everyone. My next question, question 2, just a fun question to see what kind of berry fans we have. I know there's a lot more berry crops than the ones I've listed here, but these are likely the most popular. So I thought I'd stick with these ones, keep the list a little bit shorter. And this just helps us get familiar with the polls to keep this a little bit interactive. All right, lots of strawberry fans, which is great. That's the majority of what I'm going to cover today because there's quite a few pests to go through, but good to see fans of all different kinds of berries. So thanks everybody for doing that. Just end that. That's the second one. Okay, so I want to begin with a few safety notes when you start as you start your scouting season. So it's really important to keep pesticide safety in mind. Most berry crops are sprayed with pesticides regularly throughout the season. So you should keep that in mind when you're scouting. So do not enter a field until you've confirmed whether it has been sprayed, with what and when it's been sprayed. So pesticides in Canada are reviewed and registered by Health Canada, and the scientific data that is used to register the product helps to establish the product label, which is the document that outlines how the product can be legally used. And that's the information that we put into our crop protection resources, like our Ontario Crop Protection Hub. And the label specifically includes safety protocols to minimize worker exposure during and after application, and that includes the restricted entry interval. So over time, pesticide residues and vapors dissipate until they are no longer a risk to workers that are entering the field. So the length of time required varies with the product, the rate, the environmental conditions, and the type of work you are doing. The restricted entry interval is the period of time that anyone must not do hand labor in the field after a pesticide has been applied, including tasks like scouting. And REIs can change from 1 hour to several days. And it's important to note that the REI can vary with the product, crop, and type of activity. So you might have different REIs on different crops. And the REI can change. So if the REI is 12 hours for belief on strawberries, it may not be 12 hours on another crop, or it might change every year. So it's important to look at the label or look at our protection information every year. And that's information is on our Ontario Crop Protection Hub now. And just a note, if you're using a tank mix, if you're going into a field that's had a tank mix applied to it, use the product with the longest REI, follow that restricted entry interval. So REIs can be found on the label or on our new Ontario Crop Protection Hub. And this has replaced our previous publication 360 that used to have there used to be a print copy that would have our crop protection information. So now that's on our digital hub. And we have some training videos. If you haven't seen those training videos, we just launched this year. So it's a new tool. You can find it at ontario.ca/crop protection. And in the email I sent yesterday, there's a link to a few YouTube videos that kind of train how to use this tool and where to find that restricted entry interval information. And it will show you how to get to this page, which kind of puts that information that's important to scouts in one spot. So this is an example of restricted entry interval for blueberries. I think this is Danitol. And you can see that there's different REIs for different activities. So you should not scout in blueberries here until 24 hours after the spray, but just note that there are different REIs depending on what you're going to be doing. So if anybody isn't familiar with that, check out those YouTube videos and then just play around on the hub. And it's pretty user friendly, I think. So you can use it on your computer or on your phone when you're in the field, if you have internet service. And if anybody has questions about it, feel free to send me an email or give me a call, and I can help you familiarize yourself with it. So when you're heading out to a field, make sure you have the appropriate information on what was sprayed and when. Check and follow the REI and tell your supervisor where you're going to be going, and make sure to follow any specific directions regarding personal protective equipment. And keep in mind that miscommunication can happen. So learn to recognize the signs of spraying. So fresh tracks, a distinct smell, a sound of a sprayer, or fresh residue on the leaves. I know in strawberries it's going to be pretty clear if there's a sprayer in the field, but maybe in blueberries or maybe some raspberries, it's not quite as easy to see across the field. So if you suspect the field was recently sprayed, contact your supervisor or the grower and just confirm when this field was sprayed last. And a few last notes about scouting protocols, and these are always good practices, but especially in the last couple of years with COVID. So it's always important to either check with your grower beforehand or establish a day that you're going to be scouting so that you know it's safe to enter the field, and so that they know who is coming to their farm and what you are doing. This also helps them recognize the vehicle of who's driving in and out of their farm. Communicate via phone or email ahead of time. If there's a problem area that you want to show the grower, you can flag that area, make sure you flag it well, provide the grower with directions for them to check out afterwards. If you're scouting in a field where workers are present, try and scout in a different area from them and to maintain that 6 feet of distance. And finally, it's always a good practice to drive carefully and slowly on the farm. A lot of berry farms have on-farm markets with customers and children coming and going, or they have other activities on the farm. So keep that in mind when you're visiting the farm. Drive slowly, carefully, and keep that in mind when you're parking and how to, pull out safely and so that you're not in the way. Okay, and I want to talk a little bit about what tools you will need. So these are just a few things that I think you're going to use at some point in the summer. Hand lens is really important for a lot of the pests that I'm going to go through. Flagging tape and flags to mark your traps or any issues in the field. A jackknife or a trowel in case you need to dig up any plants and cut open the crowns. A tapping tray for Tarnish Plant Bug. So you can use like a paper plate, or I actually like using a Frisbee. Pruners for cutting raspberry canes if you need to. Parchment paper for collecting sticky cards. Collection bags if you need to collect any samples and take them back to your office or your lab or to your house to take a closer look. And then a biosecurity kit if going in between farms or fields. Make sure you have clean boots. Okay, so now to get into what we're going to be seeing in the field. First up is strawberry disorders. I mentioned that I covered diseases and root pests in the pre-recorded, the videos that are on YouTube. So you can check those out for other root and crown diseases. So most of today I'm going to spend on insect monitoring after this, and frost damage. So that's what I want to start with, since this is maybe the first thing that you're going to be seeing is winter injury in the spring. So plants with winter injury can be weakened or stunted, and they're more susceptible to insect and disease damage. So winter injury happens when the temperatures in the crown drop to -12 degrees Celsius. So to diagnose winter injury, this is where your trowel will come in handy. Dig up the affected plants and cut lengthwise through the crown. And that's where you'll see this brown flecking in this picture. And there's a comparison of what a healthy crown looks like. So if conditions are slow and favorable, plants can recover from mild winter injury. But with severe injury, you'll see the crown tissue is dark brown near the top of the crown, and plants will likely not recover from that damage. So next is frost damage, which hopefully we won't see much of this year, but depending on what stage the crop was at a couple of weeks ago, I have seen some. A couple of weeks ago we had temperatures like -5. So if there was some buds present, they might have been damaged depending on what kind of frost protection was available. So strawberries often bloom before the last spring frost, especially day neutrals and June bearing fields where row covers were used to bring them on early. So open blooms are tolerant to cold injury to 1 degree below Celsius. And tight buds are tolerant to -5.5. So it depends what stage the crop is at. And if plants aren't protected and the temperatures drop to these like -1 or -5, depending on the stage, you may see some frost damage, which includes these brown or black pistils in the center of the blossoms, or crinkled leaves, or the tips and edges of the leaves may look water soaked, like this picture in the middle. So no fruit will develop from those damaged blooms, or the damage might just result in damaged fruit and misshapen fruit. So growers will use row covers, overhead irrigation, or both to protect strawberries from cold temperatures. So here row covers were used to bring the crop ahead, but they kept it on or put it back on if needed for frost protection. Later, if the temperatures dropped further than that critical temperature, the grower could run irrigation through the night on top of the row covers, or they could also run irrigation without row covers, just depending on what their strategy is. So growers have needed to use some form of frost protection like most years. And so because I'm sure many of you are aware, we have had some really cool spring temperatures like a couple weeks ago, like I mentioned. So here's a picture of after a night of frost protection. So they were using irrigation to protect these buds that were in the crown. And then here's a picture afterwards that I took a couple days ago. This is a different field where the blooms were present, and now there's some frost damage. You can see the black center of that blossom there. Just another issue that you might see is herbicide injury in the field. So that can include a variety of symptoms like leaf yellowing and mottling, speckling, necrosis, leaf cupping, or that interveinal yellowing. So if you suspect herbicide injury, look at the pattern of damage in the field and the pattern on the plant, like is it new leaves or older leaves, and ask the grower what herbicides have been applied to the field. And we have more information on diagnosing herbicide injury on Crop IPM. That's our IPM website. And I sent information from our weed specialist, Kristen Obeid, to you as well. And in this picture here, Sinbar damage is on the left and injury from glyphosate is on the right. Okay, now I'm going to move on to insect pests. So the strawberry clipper weevil is the first pest of the spring. You should begin monitoring for clipper weevil when buds are emerging from the crown, which has been a late spring, but we're starting to see buds in the crown. And I expect with the warm weather, they might move quickly. So clipper weevils will start moving into the field when temperatures are above 16 degrees. And in these pictures, you can see the adult weevil on the top left. They're about 2 to 3 millimeters long, and they have a reddish to brown long snout. And then the damage is caused by the adults feeding and laying eggs. The adults feed on the pollen in the buds and the bloom, and they leave these round holes that you can see right there. So then they lay eggs in the buds, and they girdle the stem below the bud that you can see in these pictures right here, where that clipped bud, and that causes the bud to dry out and drop off. There you go. There's that clipped bud. So to monitor for clipper weevil, begin monitoring shortly, probably now in the early spring when buds are emerging. They overwinter in protected areas, and they migrate into the field from overwintering sites. So you'll see them first along field edges. Weevils can be found on blooms and blossom clusters, but damage will be easier to find versus the actual adults. And damage occurs until all flower buds are open. And there's only one generation a year of clipper weevil, and they won't damage bloom once the bloom is open. So because they move in from the edges, sometimes they can be controlled with just a spray of the outer edge of the field. So some of your monitoring is going to be focused on the outer edge. So as I mentioned, we're going to start monitoring soon. And you can use a frame to measure a 2 by 1 foot area and count the number of clip buds inside that frame. So check for clip buds in 5 locations and focusing on the edge of the field. The threshold for clipper weevil is 13 clipped buds per 2 feet of row. And that's if your sampling is biased towards the edge of the field. So check older fields and sheltered areas for the first signs of damage. And check the field edges near woods, brush, or other overwintering sites when you're monitoring. And then when you return to the field to assess the efficacy if the grower sprayed, or you want to see if there's new damage, make sure to only count the new buds. Newly clipped buds will still be green with only a little bit of brown, while older buds will be fairly dry and brittle. So monitor for clipper weevil continuously every 3 days until the end of bloom. And then assess if there's a need for additional control. Okay, moving on to Tarnish Plant Bug. This is a major strawberry pest, and it's also a pest on a wide variety of other crops, including raspberries. So Tarnish Plant Bugs attack berries and bloom. This is a pest as long as bloom and green fruit is present in strawberries. So in the spring, in June bearing strawberries, and season long in day neutrals that have a continuous bloom period. There are 5 instars of nymphs that you can see on this in this bottom picture, and they range from 1 to 5 millimeters in length. Nymphs can cause the most economic damage, and then as these nymphs develop, they become darker green and start to develop dorsal spots on their back. And you can see the beginning of wing pads on these later instars, on the 4th and 5th instar. So the adult Tarnish plant bug on the top of the screen, they can fly, and they fly quickly when they're disturbed, so they're not always easy to see. And there are multiple generations a year. From in Ontario, there's 3 to 5 generations a year, so that is why it's a little bit challenging to control. They're just a pest season long. And the nymphs feed on bloom and young green berries, and they cause this cat-facing injury. It causes this cat-facing is kind of malformed fruit and abnormal growth, or sometimes called buttonberry. So as the fruit develops, the seeds stay clustered together, and that prevents the normal growth of the berry. And here's just other pictures comparing it to other damage, because you can confuse Tarnish plant bug damage to other issues, including poor pollination or frost damage. But Tarnish plant bug injury is distinguished from frost damage or poor pollination by the size of the seeds. So the seeds on fruit damaged by Tarnish plant bug are typically fully developed, versus the seeds on fruit damaged by frost or poor pollination are variable in size and smaller in the injured area of the fruit, but fully developed elsewhere. And that's what you can see in this comparison at the bottom. So Tarnish plant bugs overwinter as adults in the leaf litter, dead weeds, under tree bark, and they move into the field in the spring. Nymphs then usually emerge in mid-May, feeding on bloom and fruit. And the symptoms of tarnish plant bug infestation are damaged fruit. So it's important to control tarnish plant bug before injury appears. And you will need to begin monitoring in the spring shortly once there's first bloom. So begin soon in June bearing strawberries and continuously again through the season for day neutrals, and continue monitoring through to the green fruit stage. And when you're monitoring, walk the field in a W pattern, tapping flower clusters into that paper plate, tray, or frisbee. And try to tap 50 clusters in a field and count the number of nymphs that fall into the plate. So although these nymphs can look similar to other pests that we're going to go over today, tarnish plant bug nymphs move faster than some of the other pests like aphids, and that can help to identify them. So the nymph, the threshold when you find the nymphs on your tray is about 1 nymph in 4 clusters. So, and make sure to monitor varieties separately if they have different bloom periods. So if you're going to monitor one block, make sure you go back later for Valley Sunset or Malwina specifically, because they have such a late blooming period and need to be managed separately. Just a few other notes. Tarnish plant bug can quickly migrate into strawberries from alfalfa. So damage can increase when hay or alfalfa is mowed. So keep an eye out when hay is being cut. And here's another look at those early instar tarnish plant bug nymphs. And this is likely what you're going to see on your tapping tray. And they don't have those dorsal spots yet. So that threshold that I mentioned is 1 nymph in 4 clusters. But there's another method, sequential sampling, which is an alternative method for monitoring. And this just helps you make a decision faster if populations are really high or really low, because it requires fewer clusters to be examined before making a decision. So instead of counting the number of nymphs per cluster, you will count the number of infested clusters. And here's the chart to help with that decision. So after 20 clusters, if no nymphs are found, you can conclude that no control is needed. But if 4 are infested, control is required. And then if between 1 and 3 are infested, you need to continue sampling, maybe 5 more before you can make that decision. And this, again, just helps you make a decision faster than necessarily tapping 50 clusters. And then just a note on using those trays or those frisbees or plate. Try to protect the tray from the wind so you have better luck at getting a glimpse of those nymphs before they move around too much. And some insects like potato leafhopper and tarnish plant bug move quickly, so just be ready to get a fast look at these. We have a short video on YouTube that, this is 2.5 minutes long, and it's a quick summary of monitoring for tarnish plant bug. And it has a good kind of comparison of tarnish plant bug nymphs to some of the other pests that you're going to see, like aphids and potato leafhoppers. So you can check that video out if you just want a quick refresh later on in the season. And there's other videos. We have 2 other videos for strawberry monitoring, like these short videos. One's for strawberry aphids and another for Spotawing Drosophila monitoring with saltwater test. Okay, moving on to the next is flower thrips, and you might also see this in your tapping trays or on the fruit or blooms. So flower thrips are these tiny, long, narrow, yellow insects, and they're less than 2 millimeters long. Some thrips are beneficial, but Western flower thrips can cause damage to strawberries. And the adults, in this top picture, have fringed wings, but otherwise they look pretty similar to nymphs. And both adults and nymphs have sucking, rasping mouth parts, and they cause damage by feeding on the developing seeds. So they can be found in blossom clusters and under the calyx of developing fruits. And in high numbers, they will cause bronzing to the seeds and berries, especially beneath the calyx, or the entire berry can become bronzed or cracked. And the seeds on the damaged berries are more prominent. So the berries are really seedy. And there are multiple generations of thrips a year. So here's a couple more pictures of thrips damage. We saw a lot of this in 2017. So we don't always see thrips damage, but some areas tend to see thrips and damage more consistently. And thrips injury can be confused with powdery mildew or cyclomite damage. So to distinguish between the different pests, look for that rusty discoloration under the calyx, like in this photo on the left, and that will help you confirm. Show my pointer out. So that will help you confirm that you're looking at thrips damage. So thrips, so one of the reasons that they're not always an issue is because they typically don't overwinter in Ontario, but they migrate to Ontario on air currents from the south. And they're attracted to white flowers and therefore strawberry bloom. So they're usually a bigger challenge in day neutrals because of that extended bloom period, or later June-bearing varieties that bloom later, maybe when the thrips are blowing in. Thrips are also a concern in greenhouse production. So pay close attention to fields near a greenhouse. And then for monitoring, gently blow on blossoms and that will encourage thrips to move out of the bloom, or check under the calyx on developing fruit for that bronzing. And you may need a hand lens to identify the thrips because they are so small. And we don't have an established threshold in Ontario, but 5 to 25 thrips on 50 berries is a suggested threshold depending on the market where the berries are going to. So 25 thrips per 50 fruit is the suggested threshold for pick your own, or 5 thrips per 50 fruit if you're shipping. And then in the Midwest US, they use a threshold of 2 thrips per blossom. So there's a couple different thresholds, but it's important to keep an eye on thrips and just monitor their levels and communicate that to your grower. Okay, so spittle bugs are found in these white spit-like froth on the plants. They're soft-bodied yellow or green nymphs, and the nymphs pierce plant stems and suck plant juices, and then as they feed, they excrete this frothy foam that you can see in these pictures. So in serious cases, they can cause distorted leaves and stunted berries if they feed excessively on them. And spittle bugs appear in late April and early May, but they're rarely actually a pest of concern. They're more of a nuisance for your pick your own customers or something that you might notice when you're scouting. So that's why I included it here. You don't need to specifically monitor for spittle bugs, but just wanted you to be aware of what you're seeing and that it's typically not an issue for growers. Okay, potato leafhoppers are another strawberry pest that you may find in your tapping tray. So potato leafhoppers damage plants by sucking sap from the leaves and injecting a toxin into the plant while they feed. So here you can see the adult on the right and a nymph on the left. Both adults and nymphs are long and narrow and kind of cigar shaped. And nymphs are smaller than adults and they walk in a distinct sideways movement across the leaf when they're disturbed. And then adults have wings and they fly away quickly. So you're rarely going to see them when you're scouting, except for maybe out of the corner of your eye when you disturb some plants. So adults and nymphs suck sap from the leaves and inject that toxin, and that toxin blocks movement of water and nutrients to the affected area of the leaves. And that reduces vigor and causes the affected leaves to turn pale green or yellow, also known as hopper burn. And the discoloration is most obvious at the tip and edges of the leaf, which will curl downwards. And some varieties are more prone to leafhopper damage, such as Governor Simcoe and Jewel. So pay attention to what varieties you're monitoring. And it's likely most important to concentrate on first-year new plantings. So begin monitoring in late June and continue into August. Look for leaf curl and yellowing. Potato leafhoppers are found on the underside of these fully expanded leaves. And you'll sometimes find the empty skins of previous instars on the underside of the leaves as well. Leafhoppers are also pests of beans and raspberries and alfalfa. So strawberries and raspberries are at a higher risk if they are next to an alfalfa field after, especially after like first or second cut. So control is usually only needed again in those new fields or on raspberries. And here's just another picture of strawberry damage on the right, hopper burn to raspberries on the left, and that bright green cigar-shaped nymph in the middle. And these nymphs do look similar size to some of the other insects, but that cigar shape kind of helps distinguish, and that sideways movement. And that YouTube video I mentioned on Tarnish Plant Bug has a good comparison of these different insects that you're going to see on your tapping tray. Okay, moving on to strawberry aphids. There are a lot of different aphids that you might see, but the ones we are concerned about are strawberry aphids, and these are the most common aphids present in strawberry fields. So historically, we didn't really worry about aphids too much. They do not often build up to cause enough damage, and that damage could be black city mold from the honeydew that they secrete. But about 10 years ago, strawberry viruses became a challenge in Ontario, and most of these viruses are vectored by strawberry aphids, although some are vectored by whiteflies. So I covered viruses in one of the videos that's on YouTube, so check that out for more information and kind of images of identifying virus injury. But the important thing to know is that the strawberry aphids need to be monitored, and monitored regularly and controlled regularly on every farm every year. I know in the last few years, thankfully, we have seen that viruses are a little bit more under control and growers are able to keep their fields for a little bit longer. But I think the success there is because we've been diligent with aphid control. So it's important to keep that up, keep monitoring for aphids, and keep spraying for aphids regularly to make sure that viruses stay under control and we can continue to have vigorous fields. So adults can be winged or wingless. And then here is how you can distinguish strawberry aphids from the other aphids that you're going to see. Strawberry aphids have these knobbed hairs on their body. So you'll need a microscope or a hand lens to identify these knobbed hairs likely and distinguish them from other aphids. But most of the aphids that you're going to be seeing in the field are strawberry aphids. About 80% of aphids in a strawberry field are the strawberry aphid. And then when you're seeing these aphids in the field, they move slowly and they have 2 long antennae and 2 cornicles protruding from their abdomen, which you can see there, which will help distinguish them from those other insect nymphs. Strawberry aphids prefer the young succulent growth of strawberries, and they're found on the underside of new developing leaves. So on this picture on the right, that's where you're going to be picking out of the field to look for aphids. And that's a winged form on the left. And when these winged forms are present, this is when the aphids fly to new fields and spread virus. And that's the stage we're trying to control so they can't spread virus from, potentially an older field to a new healthy field. So they need to be controlled before those winged forms develop. And we don't have a specific threshold in Ontario, but you need to monitor populations weekly. And then when you notice that the populations start to increase, that's when it's time for an insecticide once that population starts to jump. So roughly check the underside of 60 new folded leaves throughout the field. So pick out these young folded leaves and make sure you kind of you're checking the underside, but look at each of the leaflets on that leaf and look for aphids. They can be a little challenging because they're green and they hide in well with the strawberry leaves, but that's where a hand lens can come in handy. Usually when you see 15 aphids out of those 60 leaves, that's when control is necessary. And typically control is going to be necessary every 3 to 4 weeks or a few times throughout the season. But make sure you're monitoring throughout the summer, and I am starting to see aphids in the field now. Okay, I have 2 mites that I'm going to go over today, the 2-spotted spider mite and the Cyclomite. If you're scouting other crops, you're likely familiar with 2-spotted spider mites. They feed on a lot of broadleaf hosts, including strawberries and raspberries. And they're pretty small mites, about 0.3 to 0.5 millimeters. And they have 2 dark spots on their backs. And these small mites are best seen with a hand lens in the field. And all stages, the adults, eggs, and nymphs can be present at one time. And they do not cause damage every year in strawberries, but they should be monitored for every year, especially in day neutrals, because outbreaks are not always predictable. So overwintering adults are orange to reddish color, and then later nymphs and adults are clear or white with 2 dark spots on their back. And like their spider cousins, spider mite adults have 8 legs and can spin silky webs. So they feed on plant sap, causing this flecking or stippling that is visible on the upper leaf surface, giving the leaves a bronzed look. And then high populations can cause fruit and sepal bronzing as well. And you might see webbing on the lower leaf surface. So the conditions that are most conducive to mite development are hot and dry conditions, because 1 generation from an egg to an adult can occur in 10 days in warm weather or 3 weeks in cooler temperatures. So you can see how the population can increase quite a lot depending on the conditions. And there are multiple generations a year. So some strawberry varieties are very susceptible to spider mites, so make sure to know which variety you're scouting and scout varieties separately. Other factors that can lead to high mite populations would be a lot of nitrogen, so plants high in nitrogen, drought stress plants, hot and dry weather, and then certain pesticides are harmful to natural beneficials, and that can lead to an increased kind of like a mite outbreak. So begin monitoring before bloom and continue after bloom on June-bearing strawberries and all season on day neutrals. Look at the lower leaf surface and then use a hand lens or a microscope to identify the mites. And you're looking for webbing, adult mites, nymphs, and eggs. So collect 60 leaflets. That's one of the trifoliates that make up a leaf. So collect 60 from fully expanded leaves, but avoid collecting leaflets from the oldest lower leaves. And then the threshold for control is 5 mites per leaflet indicates 25% of plants are infested, and 20 mites per leaflet indicates 50% infestation. So whether or not a problem develops with these thresholds depends on the variety, the weather conditions, the natural enemies, and the crop management practices. So use the lower threshold before harvest or on plants with other stresses. And then you can use that higher threshold after harvest or in non-bearing plants in your new fields. When you're looking for mites, you might also see beneficial mites when you're scouting. There are several predatory insects and mites that occur naturally in strawberry fields and feed on 2-spotted spider mites. And some growers will release beneficials to help control. So in this picture, you can see Phamoxis here, which is a predator and differs from the 2-spotted spider mite. It has a different shape. Its teardrop shape is amber versus yellow, and doesn't have those 2 spots of the spider mite. And I think one of the resources I sent you yesterday was helping identify beneficials. Okay, and the second mite that I want to talk about is a pest, is Cyclomite. And it's a pretty serious pest of strawberries, and they are smaller than 2-spotted spider mites. They're oval and white to amber in color, so they are pretty small. They're about 1/4 of a millimeter, and you will need a high magnification, so 10 to 40 times magnification to see these mites. And it can be hard to see them, but I'll talk a little bit about the damage because that's probably how you're going to identify this issue in the field. And they can cause serious damage to strawberries. The damage on the leaves will show up as wrinkled, distorted, toughened, and slightly darker leaves. And in serious cases, leaves can be stunted and crinkled and kind of compact in the center of the crown, so you have like a smaller plant. Your fruit will be small and bronzed with prominent seeds again, so maybe that makes it challenging to distinguish from other damage I mentioned with thrips. So you'll need to look for the mites to confirm the damage. And if cyclomite is left uncontrolled, they can prevent new growth and severely affect fruit quality. Cyclomites usually build up over the years, so typically they're a bigger pest in perennial June-bearing fields, but I have also found them in day neutral fields. So be aware of these mites wherever you're scouting strawberries. So because these mites are so small, identifying them is challenging. So it's easier to identify the damage. So hopefully in these next couple slides, I can help you become familiar with it. So you're looking for leathery new growth, kind of off-colored stunted plants. The petials are shortened and the leaves are smaller. So in this picture in the middle, you can see that like that plant here, where's my picture? You can see right here that plant looks off and it doesn't look healthy compared to, like, this is a nice, healthy, fully expanded leaf. And then down here, you know, these berries are looking, these green berries are looking bronzed, and the plant is looking stunted and it's compact in that crown. So it's not looking very healthy. And that, when I see that, I would think Cyclomite, and then you're going to pull out these young leaves. So that's the damage that you're looking for. And you can see this cracked and seedy berry. So that's a pretty serious infestation. And this picture again, here's like, you can see how the leaves look off compared to these healthy leaves right here. So that's a pretty good comparison. You have these wrinkled, toughened, sometimes I would describe them as crispy leaves. So that's what you're looking for to identify Cyclomite damage. So smaller leaves are going to be crowded in the crown, and if mites feed on the blossoms, the flowers can die and the fruit will become, or the fruit can be small and bronzed. So at this point in the spring, you might see healthy plants, but once it warms up and the plants start growing rapidly, which could be happening in the next week, you might see that plants infested with Cyclomite fail to grow as well. And that's when it becomes clear that Cyclomites are present. So, you know, when you're standing in a field and you can see that there's a kind of an area that doesn't look as healthy, or those plants aren't growing compared to like healthy other well-growing plants, that's when I would head there and look for Cyclomites. So begin monitoring when new buds emerge from the crown and continue until harvest. And then monitor again after renovation. Mites are most active during those periods, so May and June, and then late August and September. And so therefore at those times you're more likely to see leaf distortion. So walk a large portion of the field, but again, you're looking for areas where those plants are stunted and the leaves are distorted and crinkled. And then once you find that damage, you're going to confirm that it's Cyclomite by pinching out the newest leaves in the crown, and then open them up and use a good hand lens or a microscope to look for the mites inside that folded leaf. So in this picture here, there you go, you can see that this is a folded leaf, one of the youngest leaves. Pinch it out and then open up those leaflets and look for the Cyclomites at the base of that leaflet. So right here, you can kind of see that looks like a pile of salt. So that's what Cyclomites and eggs look like. So, a couple more bullets. So it's important to scout your newer fields first, but expect to see problems in older fields. But you're going to start in those newer fields because you don't want to spread anything from your older, potentially infested fields to your newer fields. So we don't have any defined thresholds for Cyclomites. The most practical method is to look for symptoms and verify suspected problems by looking at those new leaves. And then if you find it, my recommendations are to spray or manage that entire block, or at least that entire variety. Because they are so small, it's sometimes it's often hard to identify them until there's damage present. So once you see it in one part of the field, I would spray that entire block. So that kind of inform how you're going to monitor. So monitor different blocks separately, or maybe different varieties. And here's another close-up of what you may see with a hand lens. It looks like piles of salt in the base of that leaf. And they are one of the more serious pests because they're so small and hard to identify, and because there's limited control options. So in 2018, we found Cyclomite damage in a lot of June-bearing fields that we visited because of a change in the products we had available. So it's just very important to keep an eye out for this, look for distorted growth, crinkled growth, and then confirm by pinching out those young leaves. And the earlier we are with identifying Cyclomites, the more successful we will be with controlling them. Okay, that is a lot of information, so maybe take a change of pace. I have another poll for us. We'll do question 3. Okay, what am I? Okay, let me launch this poll. Okay, so I have a few clues. So what am I? I'm found on the underside of young. Oh, hang on. I guess I got to do the clues first. Okay, I'm found on the underside of young folded leaves. I move slowly. I have 2 cornicles, and I have knobbed hairs. Okay, now let me relaunch that poll. Okay, so am I a tarnished plant bug nymph, potato leafhopper nymph, cyclomites, or strawberry aphid? Okay, and I'll give maybe 30 seconds to do this. Okay, that's 30 seconds. Thanks, everyone. Okay, and it is, let me show the results, yeah. It is a strawberry aphid. So, yeah, potato leaf hoppers don't have those 2 cornicles or the knobbed hairs, but you are right that you are going to see potato leaf hopper nymphs on the underside of leaves. But for potato leaf hopper nymphs, you'll see them on the underside of fully expanded leaves typically, versus aphids, you're looking at that young succulent growth. And so those young leaves that you're pinching out, those folded leaves, that's what you're going to look for aphids and cyclomite. But for cyclomite, you're looking at, like, you're opening those leaves and looking at the inside. And then for aphids, you're looking on the underside of those young folded leaves. Okay, one more question. So what causes these symptoms? Flecking and stippling on, well, it starts on lower leaves and then it could move up to younger leaves. And this pest is found on the underside of leaves, and you might see webbing on the underside, and it causes, again, that bronzing on the upper surface that you might see when you're scanning the field. Maybe this is a bit of a tricky question. Okay, so is this from 2-spotted spider mite, thrips, or cyclone mite? Again, I'll give it 30 seconds. Okay, and the answer is 2-spotted spider mite. Thrips are you're gonna see more damage on thrips on the fruit. So you're gonna see the bronzing on the calyx, on the lower side of the calyx of the berries. And then spider mites are causing this flecking and stippling on leaves and kind of like bronzing when you're looking at it. And then with the spider mites, you'll see that webbing underneath. And then versus thrips, you'll see thrips in the bloom and on the fruit, and bronzing on the calyx of the fruit. Okay, thanks everybody. So just to summarize a quick review of the different pests that I went through. Aphids, so just this is a comparison for aphids and tarnished plant bug because they are fairly common pests. So aphids are slower moving. You find them on the underside of young leaves, and they have those 2 tailpipes. And then tarnished plant bugs will be moving quickly on your tapping tray. They're bright green, and they don't have those 2 tailpipe cornicles. And then for a couple other nymphs that you're going to be seeing, I mentioned spittle bug. They're going to often be associated with spittle, like the frothy spittle on the plant. Plant bug are moving quick, and then potato leafhoppers are cigar shaped, so long and narrow, and they move quickly as well, but they move in a distinct sideways motion. But they are found on the underside of fully expanded leaves. Okay, I am going to pass it off right now to Katie Goldenhar. She is our pathologist with Omafra. She's our horticulture pathologist, and she's going to speak to us about a new disease in strawberries. So Katie, can you share? Are you good, or have I done that yet?
So you're still sharing. Yeah, let me try. Okay, looks good.
Close my
Let me know if you have any issues seeing
that. Nope, that's perfect.
Okay. Okay, thanks, Erica, and good morning, everyone. As Erica mentioned, I'm the horticulture pathologist for Omafra, so I'm based in Guelph. And so I cover diseases of horticulture crops. And so this one is specifically an issue for strawberries that's come up in the last few years. So I've been working with Erica on this, and we thought it would be a good idea, since you guys are all going to be out in berry fields, to give you an update on this disease and to know what to look for, because it is new to Ontario. So it's a mouthful. So it's Neopetropsis. And so I just say it with confidence and move on. I've also mentioned that it's referred to as Pestalosa or Neo, so it's kind of referring to the same thing. But it's a new and aggressive species that has been identified in Florida. So it was identified in Florida in 2017 and has since been reported in multiple states, including neighboring states to Ontario. And in October of 2020, we did find this in Ontario. And so it can cause crown rot and fruit rot, as well as leaf blighting. So it can infect all parts of the strawberry plant. And it is more prevalent in some cultivars than others, but we're not entirely sure what its preference is for our northern cultivars. A lot of research has been done in Florida. And so we're slowly learning how this disease behaves in more northern regions, but we do know it is able to overwinter. And so the really important thing is catching this disease early, because it's really hard to manage once it's established in a field. We have no products that are registered for control, and long-term sustainability of strawberries is thought to come from cultivar development where there is resistance to this disease, but at that point that's not available. So there's some pictures there. We'll go over what it looks like on all parts of the plant. And kind of some of the lookalikes that you might see. As if you've been in a strawberry field, there are quite a number of diseases. And so it's important to kind of be able to try and distinguish between them. So on the foliage, what you see with Neopetropsis is small leaf spots with a light center and slightly darker border that expands and becomes irregular in shape. And that's particularly when it's wet and warm, you'll get some severe expansion and leaf blighting. But what you can see with these tiny lesions here is they kind of have a tan center and a darker brown outer border. And so that's pretty characteristic of this disease. You can see here, this is from our first detection in October. You have that tan center, the darker outer border, and that then it starts to become more irregular in shape and surrounded by this purple reddish hue. And then you can see here, there's another picture. This is from Georgia. You can see this is quite irregular. This is quite advanced, but it starts from this little small brown lesion and then spreads from there. So some lookalikes that you might see during this season is leaf scorch. So that's this one here. And so a big thing with leaf scorch is that you're not going to have that really circular lesion with the tan center. They're more blotchy, they're more purpley. And so that's the difference between leaf scorch and Neopetropsis. Another one that's a lookalike is common leaf spot. And so with common leaf spot, what you see is typically a purple border around a light tan center. And so remember with Neo, it's a darker brown border outside of a light tan center. And so this one is purple. And so this one is able to distinguish that way. The most, I think, maybe the hardest to differentiate here is Phamoxis. And so Phamoxis leaf blight causes this blighting. Mainly it starts at the tips of those leaves and causes this kind of V shape. And so when those Neopetropsis, I'll go back, when you have that severe blighting, they also kind of do that same similar symptoms. And so that can be challenging when they're more advanced, but with Neo, you can have lesions in the center of leaves, and they do normally start as these circular brown lesions. But Phamoxis is another common one in Ontario that is a lookalike to Neopetropsis. So as I mentioned, it can affect all parts of the plants. We're particularly concerned about the fruit rot and the impact that that may have, as that's the marketable part of the plant, of course. And so the lesions on the fruit are irregularly small and light brown to start. And then once they become more advanced, they expand and can have this black coating, which is actually the spores. So this would be typically during or after significant rainfall or leaf wetting periods. And then eventually that whole fruit becomes rotted and mummified. So you can see some pictures here. Down here, we have some of the earlier lesions, so the early symptoms. Then we have, it's starting to expand. You can see some of those black dots. And then when it becomes more advanced, you get a whole pile of those black dots, which is the spores of the fungus. And you can see a few other pictures here. So common lookalikes, a very common one we have here in Ontario is Anthracnose Fruit Rot. And so anthracnose starts as these kind of indented circular lesions. And when they're sporulating, they actually get this salmon colored mat that are on them. And so that's a difference where they're actually sporulating. They have these orangey salmon colored spores as opposed to the black spotting. So that's a good difference between Neopetropsis. Another common one is in leather rot, which is caused by Phytophthora. And so you can see that here in a few pictures. That is more of an orangey rot that is not so much like a distinct lesion as it is, it's close to the ground. Those spores have splashed up onto that fruit and caused this kind of rotting that is typically an orangey color. So it can infect the crowns. And so it's really hard to distinguish crown rot between multiple different fungal and oomycete pathogens. And so Phytophthora crown rot is common in Ontario, and we also have Anthracnose crown rot. And so what you see above ground is those plants start to wilt, and then eventually they can die. And that's very common among all of our crown rots. And so this one, if you're seeing significant crown rot that's not controlled in a field, is really important to send to a lab for proper diagnosis. And we'll talk about that in a second. But if you cut open that crown, you'll see this reddish brown discoloration, which again is very common among other pathogens. And if you haven't watched Erica's video on YouTube on these root diseases, yeah, I would definitely recommend, because it's good to be able to compare to the other ones. But as I mentioned, it is hard to differentiate with this new one. So signs are the actual fruiting bodies or the spores of the disease, and that's a really good way to confirm what you're seeing as opposed to just the symptoms, as those symptoms can sometimes look like other things. I say don't guess. I think with this disease, because it is new, if you're seeing something suspicious, please contact Erica or I, or get that sample to a lab and ask them specifically about this disease to confirm. But what you can see with some indications of this disease, and what we've seen, is if you're taking those leaves that have some suspicious symptoms and you put them into a sealed bag with a damp paper towel at room temperature, in a few days, what we've seen is that those lesions will produce these black dots. And that's a really good way to confirm if this is actually Neopetropsis. For fruit rot, it's really hard to differentiate from anthracnose. You could try and do the same thing, but it is a bit more challenging, and that fruit will rot pretty quickly. And then for crown and root rot, as I mentioned, you're not going to have these black dots forming. It's going to be very similar to Phytophthora and anthracnose rot of the crown. And so it's really important to send that to a lab if you have severe crown rot that's not being controlled. So quickly, just want to mention that this new pathogen really likes warm and humid conditions. And so it really thrives in our summers, especially when there is lots of rainfall like we had last August. The ideal temperature for growth is 25 degrees Celsius, but it can still grow within 15 to 30 degrees Celsius. Moisture is needed, and that can come from irrigation, high humidity, rain. I think what they've said down in Florida is that the pathogen needs around 24 hours of leaf wetness in order to sporulate. And so that is significant, but we definitely do get that in our summers. So it's spread not primarily by wind, but through splashing of water. And so this can be, again, from overhead irrigation, it can be from rain, it can be from equipment or people if you're walking through the field when it's wet. So they're kind of hitchhikers, and we'll talk about that in a second. And then we think that the long distance transport, you know, how it gets into a field, how it moves from field to field is through plant material. So, you know, asymptomatic, or, you know, there's one plant in hundreds that is infected, and that's how it's moving from field to field. And there's still lots of unknowns about our survival and spread in northern regions, but we do know that if it's on an infected plant, it's not going to die over the winter. It's going to be able to come back and reproduce again. But, you know, we do know that it's not readily spreading by air. So Erica mentioned some things about biosecurity and good practices for anything when you're scouting, but I specifically want to mention for this new disease because it's new. We found it in a few fields in Ontario when we don't want it to spread any further than it has to. You know, because they're hitchhikers, they're spores that are spread through water and not easily spread through air. It's really important when you're visiting berry farms to try and avoid walking through fields when they're wet. It's not always an option, but definitely try. Erica did mention starting with new healthy fields first. That's a good practice for anything, but that's especially for diseases. You're gonna try and not pick up any spores in those older fields and then go to a healthy field. So you're kind of starting in those healthy fields first and then moving to the older fields. Anything that's been in contact with the plants, especially if you're going from one farm to another within the same day, you really need to be cleaning and changing, you know, anything that's been in contact with those plants. So that could include boots, shoes, pants, hands, just really making sure that you understand that those spores are microscopic and they can survive, you know, on clothing. And so if you're going from one farm to another, it's really important to make sure you're cleaning properly. And so definitely avoid parking too close to the field, and consider if you're going to a known site of Neopetropsis. So definitely, you know, some growers are aware that it's on their farm. If you're going to a known site, then maybe consider cleaning your car before you're going to a new site, just especially if you're anywhere close to the field, because we really don't want to be spreading this disease. So like I mentioned, if you're seeing any suspicious samples this year, you can contact Erica or I, and we can get those to a lab for confirmation. You can also send them to the University of Guelph Agri-Food Lab for a fee. So it's about $120, and they'll get a turnaround time of about a week, but they're able to now distinguish the species. There is some older species, and so you really need to make sure whatever lab you're sending this to, you're asking that you want them to check for this new Neopetropsis species, because there are some older, less aggressive species present in Ontario. And so I think that's all I have. You can reach out to me at any time if you have questions, or Erica. Yeah, and I guess have a good season scouting. And I hope you don't see this disease, but you know, it's around, and so it's good to be aware of it and know what it looks like. So I'll stop there.
All right, thanks so much, Katie. That was great. All right, I will go back to my presentation. Just give me a second one. All right, good to go. Okay, so that was yeah, that was great. So I covered strawberry insect pests, and then thanks to Katie for covering Neopetropsis. And as Katie mentioned, we have a video on YouTube with other fruit rots and other strawberry diseases. We just thought it was important to include that one in this live portion because it's new, and just wanted to highlight it, and some of those biosecurity practices that Katie mentioned. So thanks again, Katie. I'm going to move on to raspberry pests, and then I'm going to speak about Spotawing Drosophila, which is a pest for all berry crops. So, okay, so first with raspberry insect pests, we have tree crickets. So there are several species of tree crickets affecting raspberries in North America, including the snowy tree cricket. The adults and nymphs are similar. They're pale, whitish green, slender, and they have a flattened appearance. And they range in size from 6 millimeters to 25 millimeters as adults. And the adults also have transparent wings. And the damage they cause is mostly to the cane when they're laying eggs in the primocanes. And that results in weakened canes, premature dieback, and an increased risk of cane disease and winter injury. Check the chat. There we go. Okay. So females chew a hole in the primocanes to lay an egg. Yeah, you're seeing my right screen. And they repeat this several times. And that leaves a zipper-like scar running down the cane, and that's what you can see in these pictures. The puncture wounds from egg laying are usually in the top half of the cane, and the scar is approximately 25 to 70 millimeters long. And above the damage, that cane will be weakened. So look at fruiting canes in the spring for rows of puncture wounds, and look for nymphs and adults in midsummer to decide if insecticides are needed. So look for puncture wounds and primocanes in late summer as well. And growers can prune and remove infested canes where possible. Okay, now the raspberry crown borer. This raspberry crown borer, the adults look like a yellow jacket, and that's in this, my pointer back. So it looks like a yellow jacket wasp, but that's your raspberry crown borer adult. These raspberry crown borer, they lay eggs in the underside of raspberry leaves in the summer, and they lay these eggs from late July to September. And then after hatching in September and October, the larvae will move down to the base of the cane to overwinter under the soil surface. And then in the spring, they bore a tunnel into the crown, and they spend the season and the next winter in the crown and roots. And then they emerge as adults between late July and September, the second season. So they have a 2-year life cycle. There's a few notes. And that's a picture of the larvae at the bottom. Get my pointer to work. Okay, so the damage from these raspberry crown borer, the canes will become weak and spindly, and fruiting canes will often collapse before the fruit is mature, before you've harvested. The foliage can wilt and die on infected canes, and the planting will eventually thin out and eventually die. So to monitor, if you suspect crown borer, dig up the crowns, carefully shake off the soil of the roots, and inspect the crowns for larvae, frass, or tunneling. Cut through the crowns to look for the larvae. Fully mature larvae are between 25 to 30 millimeters long, but they're a bit smaller that first summer. Remember, it's like that 2-year life cycle. And they're in the, so that first summer when they're in the crown, they're a little bit smaller, only 13 to 22 millimeters long. And infested canes will be weak. They may have wilting leaves or bent canes. And if you pull on that cane, you may be able to see damage. And pulling on that cane will usually cause it to break off fairly easily. Okay, so and then there's a second borer I'm going to cover, the raspberry cane borer. And these adults, this is the adult here, this black 1/2-inch long beetle. And this cane borer lays eggs in the cane, and that causes the tips to wilt and die back. So they lay eggs 4 to 6 inches below the tip, and they chew these distinct zipper-like rings around the cane, and they lay eggs between the rings. So the larvae are legless and white, and they have a round brown head, and they bore down the stem to the base, eating the cane pith as they travel down, and they can live there for 2 years. And damage becomes more serious when the larvae starts to burrow down, and that can cause the entire base of the cane to die before the fruit matures. So you're looking for these 2 zipper-like rings, and it's pretty distinct when you see it. You'll see the tip of the cane is wilting, and then you'll see these 2, those 2 zipper-like rings. So it is pretty clear when you see that damage. Here's just another picture. So that's what it looks like with the top of the cane wilting and those two rings. So to monitor, that's what you're looking for, because you're rarely going to see the adult cane borers. You're mostly going to see this damage. And then you can cut that cane open and see if you can see the eggs inside or the larvae burrowing down. And the raspberry cane borer is not often an issue that growers need to spray for. They can often just prune out those canes to manage it, but it is important to recognize the damage that you're seeing. So next is the raspberry fruit worm. The adults are these small light brown beetles, and they feed on leaves, buds, and blossoms. And heavy damage can result in incomplete damage to those leaf buds. And then the larvae are creamy white, and they're pretty small, only 6 millimeters long. And you're often going to see them at harvest because they feed on the fruit and they cause dry, decaying berry drupelets. So to monitor for this raspberry fruit worm, look for leaf feeding around the developing fruit buds in the spring. Adults can be found by if you're shaking the bud clusters into a dish, similar to strawberries when you're tapping those clusters over a frisbee or a plate. But you can also see the fruitworm likely by walking through and just quietly observing the plants. That's an effective way to find the adults as well. So you can see the larvae in that top picture and how small it is on the fruit, and then the adult on the bottom with the clubbed antenna. Okay, and then the raspberry sawfly can be confused with raspberry fruitworm damage, but there's a few ways to tell the difference. So the raspberry sawfly leaves this clear, recognizable damage, the shredding of the leaves. The adults are small black wasps, but the larvae are more identifiable. They're these bristly green, bright green larvae in the bottom picture. So the larvae feed on foliage, causing these long holes in leaf shredding, so that leaf eventually will look skeletonized in severe infestations. Again, likely in a vigorous field, in an established field, raspberries can easily tolerate this feeding damage, but in serious infestations, it's important to notice what you're looking at, and they might need to be controlled. So here's a wider view of those skeletonized leaves. There's one generation of raspberry sawfly per year. And I mentioned it can be confused with feeding damage from raspberry fruitworm. The difference is that fruitworm beetles feed on leaves and buds on the fruiting canes, while sawfly larvae tend to feed on new primocane leaves. And you're often going to see those green bristly larvae on the underside of those primocane leaves for raspberry sawfly. Okay, now a couple diseases for raspberries, including anthracnose. Anthracnose is a serious disease on black and red raspberries. And anthracnose can infect the canes, leaves, petioles, and fruit. And the first infection usually occurs on those new primocanes, that new growth in the spring. And the lesions on primocanes are small and gray with purple borders. And then on second year floricanes, the lesions are small, rough, sunken cankers. So here's the damage on your primocanes and on your close to your fruit. So eventually those lesions can join and expand and eventually entirely girdle that cane. And affected canes are more susceptible to winter injury, and they produce weak growth in the spring. This infection is favored by splashing rain or wind and wet weather conditions. And then young green tissue is more susceptible to infection in the green fruit stage. And infected fruit will have shrunken brown drupelets and dried berries. And infected canes have lesions at the base of the cane early in the season and then further up the cane later on in the season. So to monitor, look for lesions on overwintering canes. And then from spring through to bloom, check primocanes and developing laterals for that purplish spots with the gray to white centers. Okay, raspberry leaf spot causes greenish to black spots on the upper surface of new leaves. Spots progress to whitish gray with a distinct margin, and they have the shot hole center. And then in severe cases, raspberry leaf spot can lead to premature defoliation by the end of the summer or early fall. So look for these leaf spots on the leaves, the gray leaf spot on the upper surface of young leaves, and then pay close attention to plants that have had this disease in the past, and especially watch for symptoms after a period of heavy rain. And rainy weather in the spring and summer can spread the spores of this pathogen and spread the disease throughout your field. So especially monitor after rainy weather this spring. Okay, the last raspberry disease I want to cover today is raspberry crown and cane gall. So this disease is caused by a bacteria, and it infects roots, crowns, and lower stems of raspberries through wounds. And the bacteria causes the plant tissue to grow abnormally, causing this spongy wart-like galls on infected crowns and roots. The galls eventually become hard and woody as they age. On the lower canes, the galls will split open and dry out at the site of the gall. And then the galls will develop, as the galls develop, they compromise the water and nutrient uptake. So the plant will become weak, stunted, and unproductive. And then the leaves will turn yellow and dry up at the edges. So, and here's a close-up of those galls on the cane and roots. So look for deformed plant tissue. See, these are pretty distinct deformed tissue on the plant. This is pretty clear that there's an issue going on. So look for that tissue near the crown of the plants and on, especially if you're seeing that there's swelling or cracked canes, and that can indicate old gall sites. And then if plants are weak and unthrifty, dig up the entire crown and root system to look for those galls. Okay, that was a fairly quick overview of a few raspberry pests. And now I'm going to spend a little bit more time going into a bit of detail on Spotawing Drosophila, because this is the most serious pest for raspberry and blueberry growers. And it is also a pest for strawberry growers and other soft-skinned fruit growers in Ontario. So Spotawing Drosophila, or SWD, or Spotawing, is a serious pest for soft-skinned fruit growers, because these fruit flies can lay eggs in ripening intact fruit. So other fruit flies attack overripe and infested fruit, but Spotawing can attack and lay eggs in healthy, ripe, and ripening berries, because they have a serrated ovipositor that allows them to lay eggs in those healthy, firm fruit. And then when they lay eggs in healthy, ripe berries, this causes the fruit to break down, become soft, leaky, and unmarketable. And this will result in, the berries breaking down faster and not as long shelf life, and just there's a low tolerance for this. So one of the reasons Spotawing is so challenging is because the females can lay hundreds of eggs in her lifetime, and there are multiple overlapping generations a year. So they typically emerge in June in Ontario, early June, and by mid-July, populations are high enough that growers need to control them. So it's estimated that there can be between 3 to 9 generations a year in Ontario, and that activity continues until the winter. So and they become more active in July. So because they become more active in July and that population increases, they're more of a concern for summer fruiting raspberries, fall bearing raspberries, and day neutral strawberries that are being harvested during that time. But monitoring is important so that we don't miss it if we need to control it on June bearing strawberries or like early summer fruiting raspberry varieties. It's particularly difficult to control Spotawing Drosophila in certain crops like day neutral strawberries, fall bearing raspberries, and blueberries, because growers need to spray for them regularly throughout the long season, and some of those crops are being harvested for a few months, so they need to spray for them or control them that entire time, and there's a limited number of insecticides So that's why monitoring is important. If you can time that first insecticide, you know, so if you can monitor to inform the grower or inform like your spray schedule and when to start, that can either save your crop if you are spraying earlier than you intended to. That can mean that you're going to spray and protect that crop, or you can save a spray. Let's say you're going to spray a week later because monitoring has indicated spotting isn't present, and you can save that spray, start a week later, and then maybe you have an extra spray at the end of the season because we are limited with the number of insecticides. If you can extend your use later on, then maybe keep harvesting those berries a little bit later, because sometimes you can run out of insecticides, just depending on what crop and if you need to respray after a rain or something like that. So monitoring is important. And part of that is also because spotting emerges at a different time every year. It fluctuates a little bit because of a variety of factors, like the overwintering temperatures and the wild fruit that's present in the spring that allows the population to build. So we don't always know when it's time to spray. And so I mentioned that, you know, blueberries and fallberry raspberries often will have to spray for most of the season, but sometimes Juneberry and strawberry growers don't need to put a Spotawing Drosophila spray on because the season is done by the time the Spotawing population increases. But sometimes it's an earlier season, and certain varieties, like maybe late Juneberry varieties like Valley Sunset or Malwina, do need to have a control for Spotawing on them. So that's where monitoring comes in, and it's important to know when it's active on your farm. So just here's a couple pictures to recognize the damage. Oops. I'll jump ahead, there we go. Okay, so it's easy to miss on certain berries because right now, like this could look like just a little bit of damage from being picked too hard or something. Like there's just a little bruise on them, but that is damage from Spotawing. And the fruit will be unusually soft. And here's a more extreme picture where you can see that the fruit is breaking down and completely unmarketable. So on day neutral strawberries on plastic culture, when you see the juice on the plastic, that's a good hint that Spotawing Drosophila is present. And then on raspberries, you'll find juice in the cup. So someone told me that when their pickers find juice on the receptacle when they pick those raspberries, that's when they know that Spotawing is present and that that berry isn't marketable. So that's another good hint to recognize the damage. So I mentioned why it's important to monitor. And the way that you monitor for Spotawing is to identify the ovipositor on the females or the spots on the wings of the males. So that's why they're Spotawing Drosophila. These males have spots on their wings, and that's how you distinguish them from other fruit flies. And then the females have this serrated ovipositor, and that's what allows them to lay their eggs in ripe, healthy, intact fruit. So you can see that they're fairly small, like they're the size of a regular fruit fly. So it requires magnification to see the ovipositor on the females, definitely. Very hard to see in the field. You may be able to see the spots on the wings of the males in the field if it's on a sticky card or something, but that's what you need to look at to identify if what you're looking at is Spotawing Drosophila. So there's a few options on how to identify the adults. Oops, jumping ahead, or the larvae in the fruit. That includes liquid traps, which we've used for years in OMAFRA, and that's what we started monitoring for when Spotawing Drosophila first showed up in Ontario. You can also use a saltwater test, a plastic baggy test, or sticky cards. So the first two, just checking. So the first two tests are to assess the presence of Spotawing Drosophila larvae in the fruit. And this is good to know to assess your control program and the quality of your berries. But monitoring adults is also useful, and because that helps you make your control decisions especially on your first couple sprays. But first, to identify or assess the quality of your fruit, you can use a saltwater test. So you need to collect 50 ripe, healthy berries, and it's important that they are not damaged at all. because with these tests, you're identifying the larvae only, which are difficult to distinguish from other fruit fly larvae. So to be confident that anything that emerges is an SWD larvae, it's important that you only collect healthy berries so that we know that the only thing that could have laid an egg in there was Spotawing Drosophila. So you're going to place that fruit in a saltwater mixture, which is a 1/4 cup of salt to 4 cups of water, or 1:16 ratio. And then when markable fruit, cover that fruit in salt water and gently mash it up. Any larvae will emerge and float to the surface of the water. And that's when you'll be able to kind of see those small thread-like larvae on the surface. So routine assessments like this can be used to help determine how well your management program is working, when it's time to shorten your interval between sprays, or the best way to market your crop. The plastic baggie test is a similar idea to the saltwater test. You're going to use 50 ripe, healthy fruit, put them in a Ziploc bag, and place them in a warm area. And then the warm, like a window dash or on the windowsill or the dash of your car. And the warm conditions will drive the larvae out of the fruit, and they will appear on the surface of the fruit or on the inside of the bag, like you can see in this picture. So that's what you're going to be looking for if you're going to do the plastic baggie test. But again, those tests are identifying the presence of larvae in your fruit. And a lot of what we are interested in as well is identifying adult activity, which will inform your first sprays. So the traps, these liquid traps identify the adults, and they're the important tool to use to determine when to start spraying. So you need to use, if you're going to use a liquid trap, you need a lure and a liquid bait, and then you need to take the liquid contents back to a lab once a week. So there are commercial lures available, like the Sentry lure, Alpha Sense lure, and then you can combine that with water to attract Spotawing and collect them in that liquid. Then you take the liquid back to your lab, and you need to look at each fly underneath a microscope to look at the ovipositor or of the spots on the wings if they're males. So there can be hundreds of insects sometimes in that liquid trap. So it is effective. It has been an effective tool for us to use, but you can imagine it's quite time consuming, labor intensive, and not really practical for a lot of people to do on their own. So a couple of years ago, we started looking at alternatives. And one of those was developing a molecular test for Spotawing Drosophila. So that test is now available at the University of Guelph Pest Diagnostic Lab. So growers or scouts can set up these liquid traps. So again, you need a lure and a liquid base. And then you can send the liquid contents to the Guelph Diagnostic Lab. So you would collect, say you had 4 traps on your farm, you could collect the liquid contents, put them into one cup, and send that to the Guelph Diagnostic Lab, and you'll get results within a couple days on whether or not Spotawing is present or not. So if anyone's interested in the molecular testing, you can contact me or the University of Guelph Lab for more details. One of the other tools that we've been working on is sticky cards. So a few years ago, these lures became available, these solid lures that you can pair. So instead of using something that attracts the fruit like apple cider vinegar, which means you have to use a liquid trap, there are these solid lures that you can pair with a sticky card. So we've been using these the last couple of years and working on them to learn more and identify what traps work the best. And we have found good success with yellow sticky cards and a Sentry lure. So this is the Sentry lure right here. You can see it. It's just kind of like a little baggie with a kind of a gel, and that's the lure in there. So the idea with these sticky cards is that scouts or consultants or growers can check these traps once a week and identify the males on the trap in the field. So you're only identifying the males because it's too challenging to identify the ovipositor on the females, but you'll still have it'll still be accurate monitoring if you can only identify the males. So it takes a bit of time to become familiar with what you're looking at on the trap. But once you identify your first Spotawing, you'll become familiar with it and kind of be more confident what you're looking at, and it will become a faster, quick process. And this is what they're going to look like on the trap. So you can see it looks pretty clear here. You can see the spots on the wings, the golden bodies, the red eyes. And these two pictures right here were taken with a smartphone. So if you know, you can use that yourself to help like zoom in on the fly and get a better idea of what you're looking at, or you can use that to take a picture and send it to me, and I'm happy to confirm whether or not that's Spotawing that you're looking at. Nose back. So if anybody's interested in using these on your farm, please contact me, and I'm happy to help you set these up or talk to you about how to set them up and help you identify Spotawing on those cards for the first time as you're becoming familiar with what you're looking for. And here's a nice example of all the males we found on sticky cards. So hopefully you can see how clear it can be. So we recommend setting up 4 traps per farm by early June. And the important thing to remember with Spotawing is that you need to control Spotawing Drosophila when it's present and when ripe fruit is present. So you don't need to spray, growers won't need to spray if there's no ripe fruit, or if they're not harvesting any berries yet, because the insecticides are targeting the adults that are laying eggs in the ripe fruit. So you don't need to spray until those conditions are present. So that can kind of identify when you're going to set, excuse me, when you're going to set those traps up. So, but you might set them up a few weeks before your crop, put them close to the fruit in the shade, but be careful with getting them stuck, getting these traps stuck on leaves. And that's something else that we've worked on, because I do find these traps really effective and quick, and a lot easier to work with than liquid traps, but they can still be a pain when you're putting them in the field if they get, you know, like a lot of other insects caught on them, or leaves, or if they're just getting really dusty. So that's a challenge, and that's something that we've been working on. And so last year we tried using different cages to protect the traps from leaves and other bycatch. So we designed different cage designs to see what was going to work the best. And the idea is that you can just slide this cage right over. So the bottom of the cage is open, but it was very effective at keeping leaves off and large insects off. So you can see the comparison here. So this has no cage. This is just a standard sticky card in a raspberry row, and there's some leaf stuck on it, but also a lot of big insects and big flies that just make it harder to identify Spotawing and make it a longer process and not as nice to work with. And then with using a cage, you can see how clear these are. There's way fewer insects on here, and so it's easy, if there is Spotawing, it's easy to take a quick look and identify it. So we found that using a cage was just as effective as compared to no cage with monitoring to getting that first activity of Spotawing Drosophila. So we do think it's an effective solution if you want to kind of separate the trap from the leaves. The one thing that we did learn is that you need to have a cage that has space around the trap, because if the cage is too close to the trap, then that trap could still become like covered by leaves. So even though those leaves aren't going to get stuck on the trap, it's still going to impede maybe the lure, like the sense from the lure attracting Spotawing, or from Spotawing getting on that trap. So make sure that you have space, like in this cage, there's lots of space around the trap in between the cage. And this was the other one we did where the cage was very close to the trap, and that was too close, and we think it impeded Spotawing from reaching the trap. So I think these cages are effective. They just need to have space around them. So to sum up on Spotawing Drosophila monitoring is really important on your farm or in your area. So previously, OMAPRA has done quite a bit of monitoring. And then over the years, as we've learned more, we do not have as many sites. I do hope to have a few sites this spring, but it won't be as, I won't be able to cover as many areas as we once did. So monitor on your farm if you can, or hopefully if there's somebody, or if I'm monitoring the area, then you can use that to determine when to spray. So pay attention to the blog, and I'll post those there. If you're going to be monitoring, sticky cards are quick and easier to use compared to liquid traps. You need to pair them with a lure, such as a Sentry lure, Tresse lure, Alpha Sense lure. I like using traps with a dry edge. So some of these cards are really sticky, and I know that's not nice to work with when you have a busy day, especially, and you just want to check them quickly and go. So if you use one with a dry edge, they're a lot easier to handle. There's no glue on those edges of the trap. I also like using a trap with like a sticky card with a grid, and that helps orient you when you're looking at the trap to make sure that you know, just kind of like keep your place on what you're looking at, and cover the whole trap. So if you can get a card that has a grid and a dry edge, those are a lot nicer to work with. And then creating a cage with space around the traps, decrease bycatch and trash on the cards, making them easier to work with. And as a final note, when Spotawing is active and ripe fruit is present, that is when it's time to spray. So I think it's important that everybody be monitoring or be paying attention to what's going on in the area, especially in areas that are a little bit more advanced. You know, if you're in Eastern Ontario and we're starting to control for Spotawing, in Southern Ontario, it's a good indication that you'll need to start controlling soon. So watch my blog for those updates. And if you have any questions about monitoring for Spotawing Drosophila, please let me know, and I'm happy to help. Okay, so I know that was a lot of information. That is all the pests that I'm going to cover today. I'll post this on YouTube so you can revisit this if you need to. And I also have a few resources that I'm just going to go through too, so you have something to reference when you're on the road or in the field or have questions. So we have our Ontario Crop IPM website, and there's a few different crops on here, including strawberries and raspberries. And there's a lot of information on different pests in here, including many of the pests that I covered today and on the other pre-recorded videos. So you can see that has information on identifying the pest, period of activity, biology, scouting notes. I really like this often confused with feature because it gives a comparison of different pests that might that look similar to help distinguish what you're looking at. So you can see here we have a good comparison of powdery mildew and leaf scorch, and I just like seeing it side by side, and that helps to identify what you're looking at. There's also this identification key, which you can use to look through symptoms when you're, if you need help to diagnose what you're seeing in the field and you don't know what pest you're looking for. This kind of takes you step by step through to hopefully help identify what the issue is. So I encourage everybody to subscribe to the blog. It's onfruit.ca. I post regular information on berry, crop, and pest management updates. So hopefully this is helpful for scouts and consultants to watch, so that you have an idea of what we're seeing in the field and what pests are of a concern at that time. So you can subscribe to that, and you can subscribe to specific commodities if you're interested in strawberries, or sorry, if you're interested in berries and apples, for example, and you don't want to get information for all crops. Or if you're interested in everything, then you can subscribe to apples, berries, tender fruit, and grapes. So I encourage everybody to subscribe to that. We have the YouTube videos that I mentioned. So here's the link. It's probably easiest to look up the title. So monitoring for Spotawing Drosophila, or monitoring for Tarnish Plant Bug, Omafra on YouTube. And there's a video. You can see that we have these 3 videos right here. So these are the 3 like short 2.5-minute videos on monitoring. And then I have the other courses for this work, the other videos for this workshop as well on YouTube. So check that out. There is a nice website called Phytographics. So just phytographics.com, and it's by I think it was a grad student, Madeline Dowling, and she put together these excellent pictures of different strawberry diseases. So this is from Clemson University. So this is for anthracnose. Just it's nice to see a lot of different pictures of it, very clear, high quality pictures that you can check out. And there's a video on these. I think she has a video on anthracnose and botrytis. So something to check out if you want to see some different pictures. And then maybe a video on the biology of these pests. So I really like that as well. And then there's also an My IPM app, and this was developed in the States, and it includes blueberry and strawberry diseases and insects, and it includes pictures of the different pests, information on pesticide resistance, information on diagnostics, disease cycle, signs, and symptoms and management. So I think this is a nice tool like to have on your phone when you're trying to identify what's going on in the field. But keep in mind that this is an American app, so the information on pesticides is interesting because it includes efficacy, but not all the products are registered in Canada or are registered at the same rate. So make sure to use our Ontario guidelines for the Canadian labels. So if there's any other apps or resources that anybody wants to mention or they find helpful, I'm sure everybody would appreciate seeing those in the chat. Otherwise, that's it for me. So thanks very much, everybody. Here's my contact information. You can email, call, or text me anytime. I'm happy to help with any questions that you have about berry pest scouting or pest management. So thanks again, everybody. I'll post this on YouTube and send you a link on where you can find this information. All right. Thanks, everyone. Are there any questions? I'll stop sharing. All right, if there's no questions, thanks for joining, everybody. I'll stick around for a couple of minutes in case anybody wants to ask anything. Otherwise, have a great season, and thanks for joining on such a beautiful day. Bye, everyone. All right, no more questions, then we'll head out. Thanks, Matt, for monitoring the chat and recording this. I think you can end the recording if you haven't already. Oh, there we go. Stop recording.